Hello dear friends, myself Dr. Asha Narhe, Assistant Professor, Department of Botany from Changu Kana Thakur College, New Panvel. Today I am going to give a lecture on a cell cycle, which is a topic of cytogenetics. So how the cell perform their division from a single cell, it goes to divide into the two daughter cells in case of somatic cells, and in case of gametes, so there are four cells, daughter cells are produced. So what is a cell cycle? So the sequence of events by which a cell duplicates its genome, synthesize the other constituent of the cell, and eventually it divides into the two daughter cells is termed as a cell cycle. So the first of all discovery of cell cycle was by Howard and Pillay from in a 1953. So you can simpler word you can define the cell cycle as the cell cycle is the series of event involving a growth and division of the cell from the time of its formation to own to the its own division into the two daughter cells. It is nothing but the cell cycle. So there is a generation time, a duration of the cell cycle, that is the period between the two successive cell divisions, or from the beginning of one cell division to the beginning of the next one. It is called as a generation time, and there are factors which affect this generation time. So whether the type of a cell, whether it is a bacterial cell, whether it is a mammalian cell, whether it is a plant cell, so this particular first factor is affect on the generation time. So another factors as like the environmental factors such as oxygen supply, the food availability for the cell growth, and the temperature in which the cells are now living. Then examples of generation time. So the first of all bacterial cells. So every bacterial cell divide after every 20 minutes. So their reproduction time, or you can say cell cycle time, is only 20 minutes. In case of yeast cell, the cell divide approximately after every 90 minutes. But in case of mammalian cell, so our mammalian cell divide roughly once in a 24 hours. Then in case of the plant such as the onion, so the onion root tip cells may take around 20 hours to complete the one cell cycle. So from the one cell, when this particular, it is about the prokaryotic cell, that is a bacterial cell, so it goes through the particular interphase and then mitotic phase. So from the one parent cell it goes to divide into the two daughter cells, and their generation time or the cell cycle periods is differ in a different cell type. Then the particular cell cycle divided into the two main phases. First one is interphase, which is a very long and it is a non-dividing phase, and the second, which is a short one, or it is also called as a mitotic phase, and the division of the nuclear and whatever the cytoplasm is there in a cell, it divides into the M phase. Then first of all the interphase. So interphase is nothing but the preparatory phase between the two successive cell divisions. It is also known as a non-dividing stage, and this particular interphase is sometimes mistakenly called as a resting period, but actually it is not a period of any kind of rest. In this particular period, in this duration, in an interphase, cell actually synthesize the nuclear and as well as cytoplasmic content, and it grows to divide into the two daughter cells. So interphase, again, the particularly what kind of functions occur in the interphase. So the first of all synthesis of the nucleic acid such as a DNA, RNA, along with the other nuclear proteins such as the histone proteins. Then the duplication of the centriole in case of animal cell, synthesis of energy rich compound of mitosis and a protein which are the needed for the spindle formation, and at the end of the interphase. Then this interphase takes up to the almost 70 to 90% of the total time of the cell cycle, lasting for 10 to 20 hours. So interphase, it is divided into the three stages. First one G1 phase, second S phase, and third one is a G2 phase. And G1 phase is called as a first growth phase, or it is also known as a post mitotic gap phase. Then S phase is nothing but the synthetic phase, where the actually nucleus or the whatever the nuclear content RNA and DNA they get duplicated. It is called as a synthetic phase. Third one, that is a G2 phase or gap 2 phase, or it is also known as a pre-mitotic gap phase, and second, growth phase. So in interphase, almost about 70 to 80% of the time period which is taken by the interphase, and that interphase differentiated into the G1S and G2 phase. But sometimes after completing G1 phase, some cells are go to the G0 phase, which is a quiescent phase. I will explain laterally. So the first of all, what is the G1 phase? So it is a first growth phase in which the particular cell grows in a size and synthesizes the proteins and the RNA. But in this particular nucleotides, so whatever the synthesis phase is there after completing G1, so for synthesis of the DNA, so nucleotides are required, and all that nucleotides ATP, GTP, CTP and TTP, they are synthesized in a G1 phase. Then cell organelles, which are the chloroplast, mitochondria, Golgi bodies, so all these cell organelles are increases in number, but DNA synthesis does not occur at this stage. Only the nucleotide synthesized in a G1 phase. But some cells, they get tired and they may go into the G0 phase, which is also called as a resting period, or it is also called as a quiescent phase. So if that particular cell not going to divide further, or some of the cells after completing their resting period, they once again restart the cell cycle. So once the cell passes through the G1 phase, it enter into the G1 phase. But from G1 phase to G2 phase, there are some checkpoints which is called as a G1 checkpoints. So cell having itself a checkpoints, which is a cycling dependent kinase proteins are there, which check whether the cell is good or not, whatever the nuclear content is good enough for the next generation. If there is any kind of misconnection or the mismatch, so that cell does not go into the S phase. It doesn't enter into the S phase. After finishing G1 phase, cell goes to the S phase, and where the actually DNA replication takes place along with the synthesis of the nuclear proteins such as a histone. Then the particular chromatin fibers which duplicate to form the sister chromatids connected by the centromere. Then chromosomes carry a duplicate set of genes. So where the particular in a G1 phase, so the cell having only a single set of a gene. But in case in the phase of S phase, synthetic phase, the particular chromosome get duplicated, and that's why it having a duplicate set of a gene. So the two sister chromatids which are the bound by the connectors called as a centromere. In case of animal cells, centrioles in the centrosome duplicate, forming a two pairs of the centrioles. Then next one, that is a G2 phase. That particular G2 phase, in a G2 phase, proteins for a spindle and the aster formation are synthesized, RNA synthesis continues, and cell organelles multiply, as well as ATP molecules are the synthesized and stored for the mitotic phase. So after finishing DNA replication, this particular cell goes into the G2 phase. Again from S1 to G2 phase, there are again some checkpoints, because if the checkpoints are not there, that particular cell can be grow immediately, it can divide unlimitedly. So for the limitation, because it again goes for the cancer. So that's why cell having their checkpoints such as a G1 checkpoint, S2, G2 checkpoint, and again G2 to M checkpoint. So the cancer not be occur. Then next particular second phase of the cell cycle. So around 70 to 80 particular cells, 70 to 80% of the particular time which is taken by the interphase, then cell goes into the mitotic phase. So the mitotic phase begins after the interphase and includes the nuclear division, which is called as a karyokinesis, and that karyokinesis then followed by cytokinesis. Okay, so you can see here, so all finishing by this particular interphases, that particular single cell goes into the M phase, and M phase again there are mitosis or you can say the first of all the division of the nucleus or the nuclear compound, it is called as a karyokinesis, and after finishing karyokinesis the cytoplasm get divided into the two equal half, it is called as a cytokinesis, and that karyokinesis orderly distribution of the duplicate chromosome to the two daughter nuclei, and it contain prophase, metaphase, anaphase and telophase. So prophase, metaphase, anaphase and telophase, then the nuclear division takes place into the these four phases, and after completing telophase there are two nucleus formed in a cell. Then what happens, the division of the cytoplasm takes place, which is called as a cytokinesis, and then after finishing cytokinesis, that is the division of cytoplasm, it leads to the separation of the two daughter cells. So as like this cell cycle completing, and this particular specifically M phase can be two types. One is a mitotic and another one is a meiotic. So mitotic, yeah, whatever the somatic cells, or you can say vegetative cells. So vegetative and the somatic cells, from where the from single daughter cells there are a formation of two daughter cells, and this particular division takes place in the somatic cell only. In case of gametic cells, reproductive cells, there is a meiosis, and from the particular characteristic division of the reproductive cell, or the you can say germ cell, in which eventually gametes are formed from the one mother sperm mother cell, there are formation of four sperms. So it is called as a meiosis, and after finishing with the meiosis process, from one daughter cells to there are from one parent cell to there are four daughter cells produced. So as like M phase finished. Thank you.